Gene expression or gene action. Role of genes. Genes are most significant in that they exert profound influence on the multiple aspects of the life and development of organisms through governing the synthesis of specific proteins. In a broad sense, the usage gene action and gene expression represent the same process, namely protein synthesis. But in a restricted sense, gene action represents the synthesis of functional proteins and gene expression represents the synthesis of structural proteins. So, gene action represents synthesis of functional proteins. Gene expression represents synthesis of structural proteins. So, in general, genes govern a variety of functions for the operation of the molecular machinery of organisms. Also, they produce phenotypic traits or effects which are morphological, physiological, or behavioral in nature. Some of their major functions are the following: design the architectural plan and molecular makeup of organisms and control their differentiation and development, bring about phenogenesis, expression of visible and recognizable phenotypic traits by instructing and directing protein synthesis, then control metabolism by directing and regulating the production of enzymes, bring about genetic variation, biological diversity, speciation, and evolution through transposition, mutation, and recombination. Relation between genes and proteins. It has long been established that genes act and express themselves through the production of enzymes and proteins, respectively, and also that genes and enzymes have unusual specificity in action. To explain these facts, two hypotheses have been formulated. They are the one gene, one enzyme hypothesis and the one gene, one polypeptide hypothesis. It was Archibald E. Garold, 1908, who suggested for the first time that there is a direct link between genes and their protein products, and also that any hereditary disease which involves an inborn metabolic disorder results from the inheritance of an abnormal mutant gene and a defective enzyme. Carroll termed such disease inborn errors of metabolism. From Garold's hypothesis, it can be deduced that a particular gene governed the production of a specific enzyme or protein, which in turn brings about the expression of a particular phenotype. Garold further presumed that metabolic reactions occur in linear chains called metabolic pathways. Thus, a metabolic pathway comprises a linear series of interlinked reactions. The substrate of any intermediate reaction is the product of the previous reaction, and its product forms the substrate of the next reaction. The product of the last reaction, the end product of the metabolic pathway, brings about phenotypic expression. Every reaction in a metabolic pathway is catalyzed by a specific enzyme whose synthesis is governed by a particular gene. So, an abnormal mutation of a gene curtails the production of an enzyme and in turn blocks the metabolic pathways at a particular stage. This metabolic block results in the accumulation of an intermediate product, leading to an inborn metabolic disorder. Experimental evidences in support of this hypothesis came nearby 3 decades later when the Nobel laureates George Beadle and Edward Tatum in 1941 postulated the one gene, one enzyme hypothesis based on their classic work on the bread mold Neurospora crassa. One gene, one enzyme hypothesis in detail. One gene, one enzyme hypothesis, more precisely, the one gene, one protein hypothesis, is the concept that all biochemical processes are controlled by genes. Each gene governs the production of a specific enzyme, and each enzyme controls a specific metabolic function. The main points of the one gene, one enzyme hypothesis are the following: all biological processes in all organisms are genetically controlled. Each biochemical process involves a series of stepwise reactions. Each reaction is governed by a single gene so that there is 1:1 correspondence between genes and biochemical reactions. Each gene or every reaction governs the production of a specific enzyme, and each enzyme mediates a specific metabolic function. According to this hypothesis, a phenotypic character is the end product of a chain of genetically controlled enzymatic reactions. In such a reaction chain, a precursor molecule is subjected to a series of biochemical reactions in a metabolic pathway. In this pathway, each reaction is catalyzed by a specific enzyme whose production is governed by a specific gene. Similarly, the product of every intermediate reaction forms the substrate of the next reaction, and the end product of the reaction chain either fulfills a specific function or brings about the expression of a particular phenotypic trait. This can be graphically represented like gene A produces enzyme A, gene B enzyme B production, then gene C enzyme C production. A precursor is there, A to B, B to C, and then D end product that lead to phenotype. So there is a precursor A that forms to B with the help of enzyme A. That enzyme A is produced with the help of gene A, but B to C transformation is created with the help of enzyme B, which is produced with the help of gene B. Similarly, in case of enzyme C and gene C, then D is the end product. After that, phenotype is produced. If a particular gene undergoes mutational changes, or if it becomes defective and dysfunctional, the production of particular enzyme will be curtailed. Consequently, the reaction mediated by that enzyme fails to occur. So, the metabolic pathway gets blocked at this point. This interferes with the specific metabolic function, leading to serious metabolic disorders and diseases. Then, demerits of one gene, one enzyme hypothesis. One gene, one enzyme hypothesis is very significant in establishing a direct functional relation between genes and functions. Even then, it is much an oversimplified scheme, valid or applicable only for a subset of monomeric enzymes formed of a single polypeptide chain. At the same time, it fails to account for the production of the following non-enzyme proteins which do not mediate metabolic functions: oligomeric enzymes composed of two or more identical polypeptide chains, oligomeric enzymes composed of different kinds of peptide chains, a polypeptide having several enzymatic functions. Apart from these shortcomings, one gene, one enzyme hypothesis fails to provide a satisfactory explanation for two proved facts also, namely, in some instances, a single gene codes for more than one enzyme. Apart from this shortcomings, one gene, one enzyme hypothesis fails to provide a satisfactory explanation for two proved facts also, namely, in some instances, a single gene codes for more than one enzyme, and the production of some enzymes is controlled by more than one gene. All these problems were later on addressed and solved by the formulation of one gene, one polypeptide hypothesis by Ingram. Next, one gene, one polypeptide hypothesis. This is the concept that a particular gene controls the synthesis of only a specific polypeptide chain and not that of a complete enzyme or protein molecule. So, the synthesis of a functional protein appears to be controlled by more than one gene. This hypothesis is supported by the fact that some proteins or enzymes are heteropolymers. So, what is one gene, one polypeptide hypothesis? Concept that a particular gene controls synthesis of only a specific polypeptide chain and not that a complete enzyme or protein molecule. So, the synthesis of a functional protein appears to be controlled by more than one gene. This hypothesis is supported by the fact that some proteins or enzymes are heteropolymers formed of more than one kind of polypeptide chains. Each chain will have an amino acid sequence different from those of others, so each kind of polypeptide will be specified by a particular gene. For example, a hemoglobin molecule is formed of 4 polypeptide chains, namely 2 alpha chains and 2 beta chains. The sequence of amino acids in the alpha chain is different from that of the beta chain. Alpha chain is formed of 141 amino acids and beta chain is formed of 146 amino acids. So, alpha and beta chains are specified by two different genes and not by the same gene. In view of such facts, the one gene, one enzyme hypothesis is now modified as one gene, one polypeptide hypothesis. On the whole, one gene, one polypeptide hypothesis appears to account not only for monomeric enzymes but also for polymeric enzymes and non-enzymatic proteins. However, it too fails to account for the genes which never code for amino acids like tRNA gene, mRNA genes, rRNA genes, operator genes, promoter genes. Thus, one gene, one polypeptide hypothesis is applicable to only structural and regulator genes. According to the current concept, the genic unit will govern the synthesis of a polypeptide chain is a cistron. So the one gene, one polypeptide hypothesis is more aptly termed one cistron, one polypeptide hypothesis. Then collinearity of a gene and its product. Genes and their products exhibit extreme collinearity in their organization. Collinearity of gene and gene product is the point-by-point sequence relationship between the sequence of codons in a polynucleotide chain or nucleic acid and the sequence of amino acids in a corresponding polypeptide chain or protein molecule. What is collinearity of gene and its product? Genes and their product exhibit extreme collinearity in their organization. Collinearity of gene and gene product is the point-by-point sequence collinearity will be there. Collinearity of genes and gene product is the point-to-point sequence relationship between the sequence of codons in a polynucleotide chain or nucleic acid and the sequence of amino acids in a corresponding polypeptide chain or protein molecule. In other words, it is the close correspondence between the nucleotide sequence of the genetic code and the amino acid sequence of its translational product, namely protein. Eukaryotic split genes are discontinuous with alternating exons and introns. In such cases, strict structural collinearity is absent either between DNA and mature mRNA transcription product or between DNA and protein translation product. The most convincing elucidation of a direct link between a gene and its protein product was made first by Sarabhai and his team. Sarabhai and his team in 1964 and then Janowski and his team in 1967. Sarabhai and his associates analyzed the collinearity of genes and their products using the T4 bacteriophage, which is parasitic in E. coli. Janowski and his co-workers carried out their studies in relation to the enzyme tryptophan synthetase of E. coli. They could show that the enzyme is a tetramer formed of one pair each of two different polypeptides, especially specified by two distinct genes. This finding not only revealed the collinearity between genes and its protein product, but also confirmed the one gene, one polypeptide hypothesis. The most convincing elucidation of a direct link between a gene and its protein product, as made by Sarabhai and his team in 1964, and then by Janowski and his team in 1967. Sarabhai and his associates analyzed the collinearity of genes and their products using the T4 bacteriophage, which is parasitic in E. coli. Janowski and his co-workers carried out their studies in relation to the enzyme tryptophan synthetase of E. coli. They could show that the enzyme is tetramer formed of one pair each of two different polypeptides specified by two distinct genes. This finding not only revealed the collinearity between gene and its protein product, but also confirmed the one gene, one polypeptide hypothesis. Antisense strand is there, template strand is there in case of DNA duplex. Later, transcription will be happening. Start codon is there, translation methionine, leucine, proline, arginine, serine, glycine, polypeptide chain is formed. That is a translation product, but transcription is also happening. Then deoxyribonucleotide sequence, DNA codons, transcription, ribonucleotide sequence, mRNA codons, translation, amino acid sequence, polypeptide chain. Then mechanism of gene expression. Genes act and express themselves by governing the synthesis of specific proteins. The functional property of a protein is determined by the sequence of its amino acid sequences or residues, which in turn is determined by the nucleotide sequence of the genes which govern its synthesis. The biological information essential for protein synthesis is provided by an information coding system called genetic code. Sorry for that disturbance. And we have a video about that genetic code. It is located in DNA and mRNA in the form of nucleotide sequence. First, once again, learn about genetic code. Genetic code is simply the precisely coded genetic message or information stored in DNA and mRNA in the form of nucleotide sequence. It specifies the amino acid sequence of proteins. In brief, genetic code is the nucleotide sequence of DNA and mRNA that specifies the amino acid sequence of proteins. During protein synthesis, genetic code is first copied down from DNA to mRNA. This is called genetic transcription. In the next stage, it is transferred from mRNA to protein. This is called genetic translation. During transcription, the deoxyribonucleotide sequences of DNA is transferred into mRNA in the form of ribonucleotide sequence. During translation, the ribonucleotide sequence of mRNA is converted or translated to the amino acid sequence of protein. The idea of genetic code was first advanced by Alexander D. Downes. However, it was further elaborated and theoretically presented, and first formal model of genetic code was presented by George Gamow in 1954. Though Gamow's model was not accepted, it could serve as a pathfinder to open new avenues of thinking. The basic features of Gamow's coding system that are still valid as follows. Genetic code is degenerate. An amino acid is coded collectively by few codons. Codons are the basic units of genetic code. They are formed of base and represent the amino acids of proteins. Then codons and amino acids are collinear. That is, the amino acids in the polypeptide chain are in the same sequence as the corresponding codons in DNA or mRNA. Currently, genetic code is visualized as a complex coding system composed of a series of coding units called codons. A codon is a nucleotide triplet formed of a set of 3 nucleotides. Therefore, it is often called the triplet codon. It codes for a specific amino acids. The sum total of the codons required for the synthesis of polypeptide chain or a protein molecule is known as cistron or gene. The DNA molecule may contain many such cistrons. The 4 kinds of nucleotides of DNA or mRNA can enter into triplet combinations and form as many as 64. For example, 4 raised to 3 kinds of codons. There are only this code only for 20 kinds of proteogenic amino acids. So it becomes clear that some codons never code for amino acids, and at the same time some amino acids are coded by more than one codon. So 20 kinds of proteogenic amino acids are needed. So it becomes clear that some codons never code for amino acids, and at the same time some amino acids are coded by more than one codon. Those codons which code for amino acids are known as sense codons. Those which do not code for amino acids were formerly called nonsense codons, but they have been shown to signal the termination of protein synthesis, so they are now more aptly called termination codons or stop codons. Of the 64 codons, 61 are sense codons and the remaining 3 are termination codons. UAA, UAG and UGA are the termination codons of mRNA. Some sense codons initiate protein synthesis or translation, so they are generally called initiation codons. AUG in eukaryotes and AUG and GUG in bacteria. Initiation codons and termination codons are collectively called signal codons in distinction with sense codons. The sense codons which act together by groups and collectively code for the same amino acids are known as degenerate codons, synonymous codons or redundant codons. Of the known 20 proteogenic amino acids, only methionine and tryptophan are coded by single codons AUG and UGG respectively. All others are coded by synonymous codons. 9 of them are coded by 2 codons each, lysine, cysteine, etc. One is coded by 3 codons, isoleucine, 5 by 4 codons each, and 3 by 6 codons each. Then features of the genetic code. Genetic code is formed of trinucleotide combinations called triplet codons. The functional units of genetic code are codons. Each codon is formed of a set of 3 nucleotides and it specifies a particular amino acid. DNA and mRNA contain 4 kinds of nucleotides to code for the 20 proteogenic amino acids. If codons are presumed to be singlets, the 4 types of nucleotides can form only 4. If codons are visualized as doublets, there are 4 types of nucleotides can form as many as 16, like 4 raised to 2 codons, which can code for only 16 amino acids. However, if the codes are considered to be triplets, there would be as many as 64 4 raised to 3 codons, which are more than enough to code for 20 proteogenic amino acids and also to provide start and stop signals in protein synthesis. Thus, it becomes all the more explicit that genetic code consists of triplet codons and each codon is a trinucleotide sequence. Genetic code is universal. Until 1979, genetic code was considered universal. This means that the same code is present in all organisms. Each code specifies the same amino acid in all organisms, from the simplest virus down to the most complex plants and animals. And the same signal codons have the same function in the vast majority of organisms. The universality of the genetic code was conceived by Marshall, Cascade, and Nirenberg in 1967. It probably reflects three evolutionary aspects as follows. Like, genetic code got established at a very early stage in evolution. Genetic code is highly conserved in the sense that it has undergone little or no change in the long evolutionary history of life. Although genomes have radically changed, the different species and organisms have evolved from a very primitive to a most advanced level. Later, all forms of life have a common origin. Exceptions to the universality of genetic code may be found in some prokaryotes, ciliate, protozoans, and mitochondrial genome. These deviations are related to the termination codons. In ciliates, the mRNA codons UAA and UAG are not stop codons but are sense codons which specify glutamine. Similarly, UGA is not termination codon in mitochondrial mRNA but it codes for tryptophan, which in other cases is specified by UGG. Genetic code is commaless. All available evidence states that genetic code is commaless. This means that it typically consists of continuous series of codons with no information or indication at all to specify where one code ends and other one begins. In other words, demarcating boundary signals are absent between adjacent codons. So all the bases in a polynucleotide chain are parts of different codons and none of them serve as a boundary between neighboring codons. Hence, deletion of a single base in all probability can cause a shift in the reading frame during translation. This may result in a protein that is radically different from amino acid sequence. Genetic code is unambiguous. Genetic code is unambiguous in the sense that a code always specifies the same amino acid in all organisms. Also, it will not code for more than one amino acid all by itself, even though some amino acids are coded by more than one codon. Then, genetic code has polarity. Genetic code is polarized with a definite initiation and alteration and termination codons, not alteration, termination codons, and a fixed or definite direction for reading a message. This enables a gene to specify the same protein always. Also, it causes the code to read in a fixed direction. AUG that codes for methionine acts as an initiation codon. Therefore, the first amino acid to be incorporated in the synthesis of all proteins is methionine. UAG, UAA, and UGA act as termination codons. Then, reading a codon in the opposite direction will specify another amino acid due to the reversal of base sequence. The following are some examples. Codons UUG, forwarded reading. Leucine, but backward reading. UUG, GUC, UCG, AGG. When we forwardly read this, then leucine, valine, serine, and arginine are produced. But when we try to learn it backward, glycine, alanine, like valine, leucine, alanine, and glycine are formed. So, genetic code has polarity. Then, genetic code is degenerate or redundant. Genetic code is considered degenerate because the one amino acid may be specified by more than one codon. The collaboration of more than one codon to specify an amino acid is called code degeneracy or code redundancy. Sense codons, with the exception of AUG and UGG, which code for methionine and tryptophan, respectively, code for amino acids not individually but in groups. This out of 20 proteogenic amino acids, 18 are coded by multiple codons, but only methionine and tryptophan are coded by single codons. The degeneracy or redundancy of the code is believed to be due to the presence of more than one kind of tRNA for every amino acid and the ability of the manticodon of mRNA, tRNA to bind with the codons of different kinds of mRNAs. Then genetic code is non-overlapping. Genetic code is non-overlapping. The nucleotides of a codon do not normally overlap the nucleotides of an adjacent codon. This is not a universal phenomenon because There are instances in which adjacent codons do overlap so that the same nucleotide can serve as a part of two or more codons overlapping genome. Then genetic code has definite start and stop signals. Genetic code contains specific start and stop signals for protein synthesis. In our organisms, the mRNA codon AUG serves as the initiation codon and it codes for methionine. The mRNA codons UAA, UAG, and UGA do not code for amino acid but instead serve as chain termination codons or stop codons in genetic translation. Then codon assignment, deciphering or cracking of the genetic code. Codon assignment or deciphering of genetic code is the process which determines the exact amino acid specified by a particular sense codon. In other words, it is the assignment of or allocation of specific amino acid for a particular codon. It was the establishment of the fact that mRNA forms the information intermediate between gene and protein which paved the way for the understanding of codon assignment. Then contributions of Nuremberg in the mid 1950s. The triplet nature of the codon was almost revealed. However, it was not quite clear as to which amino acid does a particular codon specify. The first breakthrough for the solution of this problem was made by the in vitro synthesis of a polypeptide by Marshall W. Nirenberg and his team, 1961, using artificially synthesized mRNA. The use of artificially synthesized mRNA is advantageous in that its base sequence is already known. Nirenberg and his associates completed the work in two stages. In the first stage, Nirenberg, Severo Ochoa, and their colleagues made long artificial mRNAs and determined which amino acid these mRNAs could incorporate into proteins. In the second stage, specific mRNA codons were synthesized and determined the amino acid tRNA complex, so which each of them could bind. The artificial synthesis of long chain mRNAs were made easy by the discovery of the enzyme polynucleotide phosphorylase by Grunberg, Managu, and Ochoa in 1955. This enzyme differs from RNA polymerase in that it does not require DNA either as a template or as a primer for synthesizing RNA. In artificial systems, this enzyme can polymerize diphosphate nucleotides, forming a long single-stranded polynucleotide. The enzyme is found in all bacteria where it most likely serves as an exonuclease to degrade mRNA. In 1961, Nirenberg and Heinrich Mattei added artificially synthesized mRNA polynucleotides of known base sequences to the cell-free protein synthetic system of Escherichia coli and studied the incorporation of amino acids into proteins. The cell-free protein synthetic system consists of ribosomes, tRNA, and amino acids, enzymes, etc. It was produced by breaking down the cell of E. coli and then supernating, separating its protein synthesizing components by gentle centrifugation. In this process, the mRNA of E. coli gets mechanically and enzymatically degraded. The cell-free protein synthesizing system is advantageous in that it contains virtually all components except mRNA. To make up this deficit, artificially synthesized mRNA was added to it. The cell-free system has the disadvantage that it is short-living, functionally only for a few hours, and is relatively less efficient in translation. Nirenberg and his team artificially synthesized mRNAs using all bases and the enzyme polynucleotide phosphorylase. They isolated this enzyme from Acetobacter vinylandi or Micrococcus lysodictus. Nirenberg and Mattei at first synthesized poly-U messenger RNA, polyuridylic mRNA. The mRNA formed of only uracil nucleotides and added it to the cell-free system of E. coli. The reaction was the production of polyphenylalanine, a peptide chain formed of only phenylalanine. The first artificially established codon is UUU and it codes for phenylalanine. Further, Nirenberg and his team produced poly A, poly C, and poly G messenger RNAs. By introducing them to cell-free protein synthesizing systems, they could prove that AAA codes for lysine, CGC for proline, and GGG for glycine. This marked phenomenal event phenomenal event in the history of molecular biology, namely the deciphering or cracking of the genetic code or the assignment of codons for specific amino acids. Nirenberg and his associates could initiate decipher 4 out of 64 codons using homopolymeric mRNAs. For cracking the remaining 60 codons, they used copolymeric artificial mRNA synthesized using two or more kinds of bases like ACA, AAC, ACC. They could establish that if two different bases are used in equal proportion, the mRNA with 8 kinds of triplet codons would be produced, and if three different bases are used in equal proportion, 27 kinds of codons would be the result. By using artificial copolymeric mRNAs, Nirenberg could crack the codes for alanine, valine, leucine, isoleucine, glycine, proline, serine, arginine, cysteine, methionine, tryptophan, and glutamic acid. Following Nirenberg, Ochoa and his team carried out similar studies. The effects of these two groups supplemented each other with the result that the empirical formula of many codons could be revealed. However, the exact base sequence of each codon, with the exception of AAA, CCC, GGG, could not be established. In 1964, Nirenberg and Leder developed the binding assay or binding technique to determine the exact base sequence of codons. They could find that a synthetic trinucleotide mRNA codon, a ribosome, and an aminoacyl tRNA with an attached amino acid can form a complex in the presence of high molarity magnesium chloride, provided the selected codon codes for some same amino acid and is attached to the tRNA. It was also found that a nitrocellulose filter can block the passage of this codon ribosome aminoacyl tRNA complex across it. On the basis of this observations, Nirenberg and his group could determine which mRNA codon codes for a particular amino acid. Nirenberg and Leder artificially synthesized all the 64 kinds of mRNA triplet codons and attached them to ribosomes. These codon ribosome complexes were then introduced to a mixture of different kinds of tRNAs, all proteogenic amino acids, Mg2 plus ions, and the enzyme aminoacyl tRNA synthetase. With the help of the enzyme, each tRNA got attached to a specific kind of amino acid and formed an aminoacyl tRNA complex. The anticodon of this tRNA then bound to complementary codon of the codon ribosome complex. Thus, codon ribosome AA tRNA complex were formed. The whole mixture was then passed through nitrocellulose membrane. The codon ribosome AA tRNA complex absorbed on the membrane, and all other uncomplexed constituents freely passed through the membrane. The absorbed complexes were then isolated, and the amino acid and codon of each of them were identified. The base sequence of the codon of each complex codes for the amino acid of the complex. Thus, Nirenberg and Leder could determine the exact base sequence of the codons of most of the proteogenic amino acids. Then, contribution of Hargobind Khurana and his team, almost simultaneously with Nirenberg and Mathai, devised an ingenious method for determining the base sequence of the codons using artificially synthesized DNA and mRNA of a known base sequence. Using synthetic DNA, Khurana and his team succeeded in synthesizing copolymeric mRNA of known repeating sequences. These copolymeric mRNA were then used for genetic translation. The collinearity between the base sequence of the triplet codon of the mRNA and the amino acid sequence of the resulting polypeptide chain raises a clear picture about the exact base sequence of a codon that specifies a particular amino acid. Initially, Khurana and his colleagues synthesized a copolymeric mRNA of the sequence. In it, the triplet codons UGU and GUG are alternatively repeated. The polypeptide or the peptide chain formed by the genetic translation of this mRNA was found to consist of the amino acid cysteine and valine in an alternatively repeating sequence. This reveals that UGU codes for cysteine and GUG codes for valine. By the same technique, using different combinations of the four bases, Khurana and his team could determine the nucleotide sequences of the different codons which specify different amino acids. Thus, it becomes clear that the painstaking efforts Nirenberg, Khurana, and their associates could bring to light the base sequences of the codons which specify all proteogenic amino acids, and thus could compile the complete dictionary of the genetic code. They could establish that out of 64 mRNA triplet codons, 61 are sense codons which specify amino acids. The remaining 3 serve as stop codons which signal the termination of protein synthesis and genetic translation. For this outstanding contribution, Nirenberg, Khorana, and Holle shared the 1968 Nobel Prize in Medicine and Physiology. Then Wobble hypothesis. Wobble hypothesis is the proposition that the first two nucleotides of the anticodon of tRNA can pair only with the complementary nucleotides of a triplet codon of mRNA, strictly in accordance with the Watson-Crick base pairing rules, whereas the third nucleotide needs not follow any base pairing rule. So it can bind with any nucleotide in the third position of different mRNA codons. In brief, the Wobble hypothesis holds that codon-anticodon pairing follows stringent base pairing rules only at the first and second positions of anticodon, and in the third position it exhibits some freedom or flexibility for unusual or non-complementary pairing without following base pairing rules. Wobble hypothesis was proposed by Francis Crick in 1966 to explain code degeneracy or redundancy of genetic code. According to it, only the first two bases of the anticodon of tRNA pair with two corresponding bases of the triplet codon of mRNA as per the normal base pairing rules. The third base in the anticodon, however, has some amount of play or wobble that permits it to pair with any base in the third position of different mRNA codons. Thus, a single tRNA type can recognize and bind with two or more mRNA codons which differ from each other only in the third base. For example, the anticodon UCG of serine tRNA can recognize two different mRNA codons AGC and AGU. The bonding between UCG and AGC follows the usual base pairing rules, but in UCG AGU pairing, hydrogen bonding takes place between G and U, which is a departure from the usual Watson-Crick base pairing rule. Such a type of non-standard or unusual base pairing is called Wobble pairing. Wobble hypothesis also holds that a single tRNA cannot recognize more than 3 different mRNA codons, so at least 32 types of tRNA are needed to decode the 61 sense codons, excluding the stop codons. On the whole, it appears clear that a tRNA can recognize 1, 2, or 3 different mRNA codons depending upon its Wobble base, the 5' anticodon base. If the Wobble base is C, it will recognize only the codon that ends in G. If it is G, it will recognize 2 codons coding in C or U. If it is U, it will recognize the codons coding in A or G. The Wobble base is never A because it would be converted to the base eosine, which could then pair up with A, C or U. Next major topic, central dogma of molecular biology. Central dogma, Francis Crick in 1958, is the concept that genetic gene expression involves an intrinsic unidirectional information flow from the informative storage system to the final gene product through an intermediate message carrier system. In other words, it is the concept of a unidirectional flow of genetic information from DNA to mRNA to protein. According to a direct flow of information straight from DNA to protein or a reverse flow of information from protein back to mRNA or DNA is not possible. However, reverse information flow from genomic RNA to DNA occurs in retroviruses, some RNA viruses. Central dogma essentially explains the functional interrelations between DNA, mRNA, and protein. DNA serves as a template for its own replication and also for the transcription of mRNA. This mRNA undergoes translation and forms protein. Thus, according to the central dogma, DNA does not directly govern protein synthesis by serving as a template for the polymerization of amino acids, but it makes mRNA and mRNA makes protein. So, there is no direct information flow from DNA to protein, from gene to gene product. The information from DNA is first copied or transcribed to make mRNA, which serves as a template molecule and guides synthesis of a specific protein. Even though retroviruses are exceptions to this general rule, central dogma stands as a genetically valid hypothesis. According to the central dogma, biological information undergoes perpetuation and repression. Perpetuation of information occurs by the replication of the genomic DNA or genomic RNA in some viruses. Expression of biological information is a two-stage process. It involves two genetic events, namely transcription and translation. We know that. In genetic transcription, the coded biological information from genomic DNA is copied down or programmed in mRNA in the form of ribonucleotide sequence. So, transcription involves the DNA dependent mRNA synthesis. It generates an mRNA strand which is identical to the non template sense strand of the DNA duplex. The mRNA is then used as template to generate amino acid sequences and thereby to synthesize protein. Genetic translation is the transfer of biological information from mRNA to protein in the form of amino acid sequence. So it involves the conversion of ribonucleotide sequence of mRNA to the amino acid sequence of protein. Then translation of mRNA to protein is always irreversible so that the mRNA sequence used in it cannot be retrieved from protein to be used again as a genetic information. The classical concept of central dogma entails a sequential information transfer from DNA to mRNA to protein, and it involves three basic processes as follows, like DNA nucleotide dependent synthesis of DNA from DNA replication, RNA nucleotide dependent synthesis of mRNA from DNA genetic transcription, amino acid sequence dependent synthesis of protein from mRNA genetic translation. The current concept of central dogma in terms of bioinformatics holds that genome makes transcriptome and transcriptome makes proteome. According to it, genome undergoes transcription and produces transcriptome, which in turn undergoes translation and produces protein, the product of gene expression. Thus, during gene expression, there is a straight flow of biological information from genome to transcriptome to proteome. Genome is the total DNA content or biological information stored in a cell or an organism. So it represents the storehouse of biological information. Transcriptome is the total RNA copies of the active protein coding genes of a genome. Thus, it is the initial product of gene expression. Proteome is this entire repository or storehouse of the proteins of a cell or organism. It represents the final product of gene expression. Genomics, transcriptomics, and proteomics are the terms used for the study of genome, transcriptome, and proteome, respectively. So, when we try to learn about central dogma, information flow is very important. So, in case of central dogma classical concept, DNA undergoes replication, then DNA undergoes transcription to form RNA. Later, it undergoes translation to form gene product of protein. Then replication will be happening in case of DNA. Then transcription is there, which creates RNA, but reverse transcription is also there. Then RNA also undergoes replication. Then RNA editing will be taking place later. Translation occurs, which create gene product, that is protein. That is the revised concept of central dogma. But later, current concept of central dogma in terms of bioinformatics says that genome undergoes replication, genome undergoes transcriptome, that is the process of transcription. Then proteome is also produced with the help of translation. Next topic, central dogma reverse transcription. The concept of unidirectional information flow from DNA to mRNA to protein holds true only for cellular genes. So the concept of central dogma enjoyed universal acceptance only for more than 4 decades until Howard Temin and David Baltimore in 1970 discovered the retroviral enzymes, reverse transcriptase, and provided that retrovirus are exceptions to central dogma. A reversal of central dogma is found to operate in retrovirus, some RNA viruses like HIV, tumor forming or oncogenic viruses such as Rosarcoma virus. In them, information flow occurs in the reverse direction, namely from genomic RNA to DNA, but never from proteins back to mRNA. There is no known instance in which a protein species specifies a particular RNA sequence or DNA sequence. So, the translation step of central dogma is always unidirectional. Then the single-stranded genomic RNA of retrovirus serves as a template for synthesizing its double-stranded DNA copy. During this, the vRNA acts as a template and directs the synthesis of a single-stranded complementary DNA, SSC-DNA. This RNA binding DNA synthesis is called reverse transcription or teminism. It is catalyzed by the enzyme reverse transcriptase, RNA-dependent DNA polymerase. The SSC DNA then undergoes replication and forms a double-stranded DNA, often called the DNA provirus. It undergoes transcription and forms mRNA, which in turn gets translated to form the viral protein. Then genomic vRNA, reverse transcription undergoing SSC DNA will be produced. After replication, DNA duplex DNA provirus. Then transcription, mRNA translation, viral protein gene product is formed. Genomic vRNA, cDNA, later DNA duplex, DNA provirus, then mRNA, later viral protein. There are some RNA viruses in which the genetic information is copied directly from genomic DNA to mRNA without the involvement of DNA intermediary. This is called DNA replication. Examples include hepatitis C virus, coronavirus, etc. In some organisms, the sequence of the mRNA is altered by a process called RNA editing. It occurs soon after the transcription of mRNA from the DNA template. So, in such cases, the DNA sequence is not faithfully used to encode the protein product. Then, next topic, prion hypothesis. The suspected role of prions in information transfer is believed to be another exception to central dogma. Prions are self-replicating and infectious subviral particles of misfolded proteins devoid of nucleic acids. The term prion is a contraction of the words protein and infection. They are nearly 100 times or more smaller than the smallest virus. They have the power to resist inactivation by procedures which may destroy nucleic acids. Prions are formally called slow viruses, but they are neither viruses nor viroids since they are devoid of nucleic acid. Prions are in fact abnormally folded normal proteins. Their abnormal folding is due to a defect in the gene that codes for the prion protein PRP. The PRP coded by the defective gene has the amino acid proline instead of leucine. This probably causes the abnormal folding of the protein. The isolation and chemical characteristics of prion protein were first successfully carried out by Nobel laureate Stanley Prusiner and his associates at the University of California. For this pioneering work, Prusiner was awarded the Nobel Prize in Medicine in 1997. They could also locate the prion protein gene on the short arm of chromosome 20 in human beings. Prions are rod-shaped structures abundantly found on the surface of membranes of brain cells, and some other cells. Each rod is formed of as many as 100,000 molecules of prion proteins, PRP. The normal prion protein is termed PRP raised to C. And abnormal, pathogenic, and infective variant produced by the mutation of the prion gene is termed PRP5C. SC as superscript. The normal PRP superscript C is a neuron-specific and membrane-associated glycoprotein present in all mammals. PRP is encoded by a host gene called the prion protein gene. Its normal wild form codes for PRPC and its mutant form is responsible for PRPSC. Prions are curious in that they are devoid of nucleic acids or genetic materials. Even then, they can infect host cells, initiate the production of new prions, and cause transmissible neurological diseases. Prions are believed to produce new prion particles by a mechanism of protein-directed protein production. This is a total deviation from the DNA-directed and mRNA-dependent protein synthesis investigated by the central dogma. The exact molecular mechanism involved in this process is not clearly understood. It has been suggested that an infective prion protein, PRPSC, can induce normal prion proteins, PRPC, to become infective. In this case, abnormal prion proteins attach to normal ones. As a result, normal proteins transform to abnormal ones. PRPC and PRPSC are almost identical in primary structures. So, the transformation of PRPC and PRPSC involves only a change in molecular conformation. This change, in turn, is believed to result from the interaction between the misfolded PRPSC and the normal PRPC. This interaction induces PRPC and to refold in a wrong way to transform to PRPSC. Soon they dissociates. Soon they dissociate and repeat the process with other protein particles. This leads to a cascade or chain of reactions, producing abnormal prion proteins in geometrical progression. One produces 2, 2 produces 4, 4 produces 8, and so on. The large number of abnormal prion proteins thus produced clump together in a brain causing neurological diseases. Prions are rod-shaped structures found on the surface of membranes of brain cells and some other cells. Each rod is formed of as many as 1,000 molecules of PRP. Prions are curious in that they are devoid of any genetic material, DNA or RNA, but at the same time, they can infect host cells, initiate the production of new prions, and cause transmissible neurological neurodegenerative diseases. They are believed to produce new prions by protein directed protein production. This is a deviation from the normal DNA directed protein synthesis. The exact molecular mechanism involved in prion production is not clearly understood. Large number of abnormal prion proteins clump together extracellularly in brain and central nervous system and cause nerve degeneration and neurological diseases. The diseases are commonly called spongiform encephalopathies since they may make the brain porous and spongy. The common symptoms include convulsions, ataxia, dementia, behavioral abnormalities, etc. So, reproduction of prion in animal cells, protein directed regulate protein production. Prions without nucleic acids infect cells, protein directed protein synthesis, new prion protein, then assembly of new prions. Later, this creates a cycle which follows like prions without nucleic acids once again. When a prion enters a healthy organism, it induces normal proteins to transform into the disease associated prion form. It then acts as a template for the misfolding of more normal proteins into a prion form. The altered proteins accumulate in infected tissue, causing tissue damage and cell death. All non mammalian prion diseases are caused by the so called prion protein PRP. The endogenous, properly folded form is denoted as PRPC, C for common, while the disease linked misfolded form is denoted PRPSC. SC for scrappy, one of the diseases first linked to prions and neurodegeneration. The incubation period for prion disease is generally long, but once the symptoms appear, the disease progresses rapidly, leading to brain damage and death. Some other aspects related to prions. Prions are the causative agents of some chronic and transmissible neurodegenerative diseases called transmissible spongiform encephalopathies or TSEs, which occur naturally in some animals such as man, sheep, goat, cattle, deer, elk, mink, cat, etc. 4 neurological human diseases and 6 similar animal diseases are now known to be caused by prions. The human diseases are, I'm not sure about the pronunciation, pardon me for that. KURU. I don't know how to pronounce this. Kuru disease. Or then Creutzfeldt-Jakob disease. Then Gertzmann, Schrossler, Schrinkler syndrome, and fatal familial insomnia FFI. Transmissible spongiform encephalopathies in mammals. this kind of diseases. Then sheep and goats, scrappy disease. Then cattle, bovine spongiform encephalopathy or mad cow disease or MCD. Then in case of cattle, feline spongiform encephalopathy or FSE. In deers and elks, chronic wasting disease. In mink, transmissible mink encephalopathy or TME. FFI or fatal familial insomnia is a genetic disease. Its main symptom is the total inability to sleep. Initially, the ability to nap slowly disappears, then the ability for a full night's sleep is lost, and finally, the patient fails to sleep at all. The syndrome usually strikes when the victim is at his or her 50s. Ordinarily, it lasts about a year and it always ends in death because sleep is to a great extent very vital. So, FFI is an awful disease, and only very little is known about how it works. Still, it is claimed that prions attack victims' thalamus, and the disordered, damaged, or malfunctional thalamus interferes with sleep. But nothing is more definitely known about why this happens and how to stop it or ease its fatal outcome. FFI is extremely rare, known in only 50 odd families worldwide. It is still a mystery and is much more similar to a less serious type of insomnia plaguing millions of people today. The animal disease are scrappy as sheep and goats, bovine encephalopathy in cattle, then feline encephalopathy in cats, then ungulate encephalopathy in ungulates, and mink encephalopathy in minks. Small semi-aquatic carnivorous mammals of Asia, North America, and Europe. Mustela, Mustela species. Then chronic wasting disease in deers and elks. All these diseases have very slow onset and progress, but their effects are fatal and devastating. They are believed to result either from the ingestion of a prion protein of infected animals or from the mutation of normal prion protein genes. The mechanism of the action of prions is only imperfectly understood, and so the diseases caused by them have no cure at present. Then bovine spongiform encephalopathy, commonly called the mad cow disease, has taken heavy tolls of cattle during 1992 and 1993 in England and some other European countries. It probably resulted from the practice of feeding cows with protein supplements contaminated with materials from infected animals. Kuru, Kuru, I don't know how to pronounce this, and was common among the people of some parts of New Guinea, probably due to the consumption of human brain as a part of funeral rites. Then Carletson Gaddis in USA was awarded the Nobel Prize in 1976 for the study of prion disease due in the tribe of Papua New Guinea. Stanley Prisner in USA, who carried out extensive studies about prions, was awarded Nobel Prize in 1997. So, if you paid attention, you learned some major aspects related to central dogma of molecular biology and about genetic code, some points related to one gene, one polypeptide hypothesis, relation between genes and proteins, roles of genes, along with that one gene, one enzyme hypothesis, later demerits of this one gene, one enzyme hypothesis. After that, collinearity of a gene and its product, mechanisms of gene expression. We tried to interpret genetic code. Then features of the genetic code, like genetic code is universal, genetic code is unambiguous, genetic code is something which has polarity. Then genetic code is degenerate or redundant. Genetic code is non-overlapping. Genetic code has definite start and stop codons. Genetic code is commaless. Such kind of points. And codon assignment, deciphering or cracking of the genetic code. How that is done. And contribution of Hargobind Khurana, Wobble hypothesis, then central dogma of molecular biology, and central dogma reverse transcription, then prion hypothesis, some aspects about prions and some diseases, then reproduction of prion in animal cells. So that's all about this topic. So that's all about this video, which explained all these topics shortly. Thank you for listening. Sorry for the disturbances and some mispronounced words. Pardon me for those mistakes. Thank you for listening.